Beneath the cathedral silence of an ancient forest, where shafts of spectral light filter through a canopy older than recorded memory, an entire civilization breathes. The forest floor, soft with centuries of fallen needles and decomposed heartwood, conceals within its uppermost few centimeters a network of staggering sophistication, a living architecture of fungal filaments, root tendrils, and chemical messengers that moves information through the darkness the way the ocean moves sound, invisibly, continuously, and with extraordinary purpose. This is not metaphor. This is the mycorrhizal web, and its existence reshapes every assumption we have ever held about plant consciousness, ecological intelligence, and the nature of life itself. The mycorrhizal network, known informally as the wood wide web, represents a symbiotic entanglement between the fine root hairs of trees and the hyphal threads of specific fungal species, most prominently those belonging to the Basidiomycota and Ascomycota divisions. These fungi do not merely colonize roots, they fundamentally transform them, extending the effective absorptive surface area of a single tree's root system by factors as large as 700. In exchange for photosynthetically derived carbon compounds, sugars, and lipids, the fungal partner delivers phosphorus, nitrogen, water, and trace minerals from distances far beyond the tree's own reach, threading through pore spaces in the soil too narrow for any root to follow, dissolving minerals with enzymatic precision no root hair could replicate. What makes this exchange philosophically arresting is its directionality, or rather, its lack of strict directionality. Carbon does not flow uniformly from tree to fungus. It flows from areas of abundance to areas of deficit, guided by concentration gradients and metabolic demand in a manner that mirrors, with unsettling fidelity, the redistributive logic of circulatory systems. A shaded seedling struggling beneath the canopy of its parent tree may receive a net transfer of photosynthate through the shared mycelial network, effectively subsidized by a neighbor it has never touched. Researchers have traced these transfers using radioactive isotope labeling, watching carbon-14 migrate from mature Douglas fir to neighboring paper birch and back again across seasons, like wealth moving through a community that has no formal economy but enforces a kind of mutual solvency nonetheless. The organisms responsible for mediating this subterranean exchange are neither plants nor animals but occupy a biological kingdom of their own, the fungi, whose evolutionary history stretches back approximately 1 billion years, predating the emergence of land plants by hundreds of millions of years. When the first photosynthetic organisms began colonizing terrestrial surfaces during the Ordovician period, roughly 450 million years ago, they did so not alone but almost certainly in partnership with ancestral mycorrhizal fungi. Fossil evidence from the Rhynie Chert, a Devonian deposit in Scotland dated to around 407 million years ago, preserves some of the earliest known land plants alongside structures that are unmistakably fungal, suggesting that the mycorrhizal alliance is not a recent evolutionary novelty but a foundational condition for plant life on land. Lignin, the complex organic polymer that gives wood its structural rigidity, is among the most consequential molecules in the history of terrestrial life, and yet it arrived in the biosphere so abruptly, in geological terms, that its appearance coincides with one of the most dramatic events in Earth's carbon history. During the Carboniferous period, between approximately 359 and 299 million years ago, the evolution of lignified wood allowed trees to grow to extraordinary heights, their trunks accumulating biomass at a scale the biosphere had never before sustained. The atmosphere grew dense with oxygen. Estimates suggest concentrations reaching as high as 35%, compared with today's 21%, and vast forests blanketed the tropical zones of a world unrecognizable to modern eyes. The critical and often overlooked detail of the Carboniferous is this: for millions of years, the organisms capable of decomposing lignin had not yet evolved. Lignin's molecular architecture, a three-dimensional, highly cross-linked lattice of aromatic phenylpropanoid units, proved resistant to the enzymatic machinery of contemporary decomposers. Trees fell, accumulated, were buried under sediment, and did not rot. Layer upon layer of incompletely decomposed cellulose and lignin compacted under heat and pressure over geological time, Eventually transforming into the coal seems that the Industrial Revolution would tear from the Earth with consequences still reverberating through every thermometer on the planet. The forests of the Carboniferous did not merely die, they became embedded in the future, waiting to be burned. The eventual evolution of white rot fungi, species belonging to the Agaricomycetes class, equipped with lactase, manganese peroxidase, and lignin peroxidase enzymes, marked the end of the Carboniferous coal-forming epoch with a quiet biochemical revolution. Once organisms capable of degrading lignin became sufficiently widespread, the carbon that had been accumulating in dead wood for millions of years could once again be mineralized and returned to the atmosphere. The coal-forming forests receded, atmospheric carbon dioxide climbed back toward equilibrium, and the great sedimentation of fossilized carbon essentially ceased. Today, white rot fungi still perform this function in every living forest, breaking down the cell walls of fallen trees with the same enzymatic elegance that ended the Carboniferous, though now operating in metabolic real-time rather than geological time. The cell walls of living wood are composed of multiple concentric layers: the primary wall, the secondary wall with its three sublayers designated S1, S2, and S3, and the middle lamella, each with a distinct cellulose microfibril orientation engineered over millions of years of evolutionary refinement to provide maximum resistance to mechanical stress. The S2 layer, which constitutes the bulk of the cell wall's mass, has its cellulose microfibrils oriented in a steep helix around the cell's longitudinal axis, typically at angles between 5 and 30 degrees. This arrangement imparts a stiffness-to-weight ratio that exceeds that of steel when normalized for density, a material property so exceptional that engineers have spent decades attempting to replicate it in synthetic composites, rarely matching what a tree assembles from nothing but sunlight, water, and atmospheric carbon dioxide. Photosynthetic efficiency in trees is a subject of persistent fascination and persistent misunderstanding. The theoretical maximum efficiency of photosynthesis, the fraction of incident solar energy that can, under ideal conditions, be captured and stored as chemical energy, is approximately 11% for C3 plants, the category that encompasses virtually all temperate and boreal tree species. In practice, under field conditions, most trees achieve efficiencies of 1 to 2%, largely because the Calvin cycle enzyme rubus-CO, responsible for fixing carbon dioxide, suffers from a profound evolutionary compromise. It reacts with oxygen as well as carbon dioxide, performing a feudal side reaction called photorespiration that wastes energy and releases previously fixed carbon back into the atmosphere. This inefficiency is not a design failure but a historical artifact. Rubus-CO evolved in an atmosphere largely devoid of oxygen, and its active site has simply never been fully reoptimized for the oxygen-rich atmosphere that photosynthesis itself created. The boreal forest, known in Russian as the taiga, forms the largest terrestrial biome on Earth, stretching in a nearly unbroken belt across Siberia, Canada, Alaska, and Scandinavia, covering approximately 14 million square kilometers, or roughly 11% of Earth's total land surface. Dominated by cold-tolerant conifers such as spruce, fir, pine, and larch, the taiga endures winters of extraordinary severity, with temperatures routinely falling below -40 degrees Celsius in its continental interior. Yet even in this apparent quiescence, the forest is not dormant in any metabolic absolute sense. Beneath the snow, soil microbial communities continue to mineralize organic matter, root systems maintain osmotic activity, and the mycorrhizal network persists in a state of reduced but unbroken function, awaiting the return of light and warmth with the patience of geological time. Among the most astonishing survival mechanisms of boreal conifers is their preparation for winter through a process called cold acclimation, during which exposure to progressively shorter photoperiods and declining temperatures triggers a cascade of molecular responses within each cell. Membrane lipid compositions shift from saturated to unsaturated fatty acids, increasing fluidity at low temperatures. Soluble sugars accumulate in the cytoplasm, functioning as cryoprotectants by depressing the freezing point of cellular water. Antifreeze proteins align along the basal planes of ice crystals, preventing their growth and recrystallization in a form of molecular watchkeeping. The needles of certain spruce species can survive immersion in liquid nitrogen, a temperature of -196 degrees Celsius, and resume metabolic function upon warming, a tolerance so extreme it approaches the physiological limits of water-based biochemistry. Chemical signaling between trees operates across two distinct physical media: the airspace above ground and the soil continuum below. Aerial signaling employs volatile organic compounds, primarily terpenoids and green leaf volatiles such as cis-3-hexanol, that diffuse through the atmosphere and are absorbed through the stomatal pores of neighboring trees. When an oak is attacked by caterpillars, it elevates its production of tannins and phenolic compounds within hours, a response that makes its remaining foliage progressively less palatable. But simultaneously, it releases a complex blend of volatiles into the surrounding air that neighboring oaks, even those not yet attacked, respond to by preemptively upregulating their own defensive chemistry. The receiver trees are not merely reacting to a chemical gradient. They are, in the most functionally meaningful sense, receiving and acting upon a warning. The phenomenon of induced resistance in trees was first systematically documented in the early 1980s, when researchers at the University of Washington, David Rhodes and Gordon Orions among them, published observations suggesting that willows and alders in proximity to insect-defoliated trees showed elevated foliar tannin concentrations without having been directly attacked. The scientific community greeted these findings with considerable skepticism, dismissing the possibility of meaningful plant communication as romanticism dressed in laboratory clothes. Subsequent decades of rigorous biochemical and ecological research have entirely vindicated the original observations, not merely confirming that airborne chemical signals from damaged plants alter the defensive chemistry of neighbors but characterizing the molecular mechanisms, receptor proteins, signal transduction cascades, gene expression changes, through which trees receive and process these chemical messages. Deep within the living tissues of a tree's cambium, the thin, actively dividing layer of meristematic cells that lies between the bark and the wood, new cells are produced throughout the growing season in a continuous act of architectural self-assembly. The cells produced toward the outside of the cambium differentiate into phloem, the living tissue that transports sugars and other organic compounds downward from leaves to roots. The cells produced toward the inside differentiate into xylem, the water-conducting tissue whose dead, hollow cells form the structural core of wood. Each year's growth of xylem cells, the annual ring visible in a tree's cross-section, records the climatic conditions of that growing season with extraordinary fidelity, creating in the body of the tree a continuous autobiographical archive of temperature, precipitation, drought stress, and atmospheric chemistry stretching back centuries or, in the case of the longest-lived species, millennia. Dendrochronology, the science of dating and interpreting tree rings, has allowed researchers to reconstruct climate records extending far beyond the instrumental period, revealing oscillations in temperature and precipitation of a precision and duration unattainable through any other terrestrial proxy. The annual rings of bristlecone pines from the White Mountains of California, cross-matched against the rings of subfossil wood preserved in archaeological sites and glacial outwash deposits, have been used to construct continuous chronologies spanning over 11,000 years, encompassing the entire Holocene and extending into the late Pleistocene. Through these ring records, researchers have traced the fingerprints of volcanic eruptions, solar minima, El Niño cycles, and medieval warm periods in wood laid down long before any human hand recorded the temperature of the air. The bristlecone pine, Pinus longiva, holds the distinction of being the oldest confirmed individual organism on Earth in terms of verified age for a single, non-clonal tree. A specimen known as Methuselah, located in an undisclosed grove within the Inyo National Forest in California's White Mountains, is estimated to be approximately 4,855 years old, having germinated around the time that the first Egyptian pharaohs were establishing dynastic rule along the Nile. The tree's survival across nearly 5 millennia in one of the harshest, most resource-limited environments in North America is not incidental to its extraordinary longevity but causally connected to it. In the thin, dolomite soils of the subalpine zone, where drought, cold, and nutrient scarcity keep growth rates almost imperceptibly slow, the dense, resin-saturated wood that results is highly resistant to fungal decay and insect attack, and the very slowness that limits the tree's annual accumulation of biomass also limits the metabolic throughput that tends to accumulate cellular damage over time. Clonal tree organisms present a different and in some ways more profound conception of biological age. Pando, a colony of quaking aspen in the Fish Lake National Forest of Utah, consists of approximately 47,000 individual stems, each genetically identical and each connected to the others through a single, massive root system estimated to weigh 6 million kilograms, making it, by mass, the largest known organism on Earth. The age of Pando's root system is uncertain but broadly estimated at between 80,000 and 1 million years, meaning that the subterranean entity from which all of these individual trunks spring has been continuously alive through the Pleistocene glaciations, the Younger Dryas, and the entirety of human civilization without interruption. Each individual trunk lives, flowers, and dies, but the root system persists, regenerating new stems from root sprouts with the unhurried steadiness of something that has learned not to depend on any single above-ground expression. Forest succession, the process by which a disturbed site progresses through a predictable sequence of ecological communities over time, is among the most thoroughly documented phenomena in ecology, yet it retains an almost cinematic quality when observed across the appropriate temporal scale. Following a catastrophic disturbance such as fire, wind throw, or glacial retreat, the first colonizers are typically fast-growing, light-demanding pioneer species. In temperate North America, species such as fireweed, aspen, and birch that establish quickly in the mineral soil or ash, fixing nitrogen, shading the bare ground, and gradually creating the conditions that make their own eventual replacement more likely. As the canopy closes and light levels on the forest floor decline, shade-tolerant species, fir, hemlock, beech, sugar maple, begin to establish in the understory, slowly accumulating height and biomass until, after decades or centuries, they overtop and suppress the pioneers that made their arrival possible. The climax forest that emerges at the end of succession, a concept that ecologists have significantly complicated and nuanced since its original articulation by Frederick Clements in the early 20th century, is not a static equilibrium but a shifting mosaic of patches at different successional stages, maintained in its apparent stability by the ongoing disturbance events, both small and large, that prevent any single community type from achieving permanent dominance. A single tree-falling event, when a forest giant is toppled by wind or succumbs to age, creates a gap in the canopy through which a column of direct sunlight reaches the forest floor, a gap that is immediately occupied by a community of light-demanding herbs, shrubs, and tree seedlings that would have no possibility of survival beneath the closed canopy. The forest is thus perpetually regenerating itself, not as a single organism in a fixed state but as a continuously self-restoring mosaic of beginnings and endings. Fire, the element most instinctively associated with destruction, is in fact the principal architect of many of the world's most ecologically diverse and productive forest ecosystems. The lodgepole pine, Pinus contorta, has evolved a reproductive strategy so thoroughly adapted to the fire cycle that it maintains two distinct types of seed cones, serotinous cones sealed with a resinous bond that melts only at temperatures above 45 to 60 degrees Celsius, releasing their seeds in the immediate aftermath of a crown fire, and non-serotinous cones that open normally in the absence of fire. The serotinous cones can remain viable on the tree for decades, accumulating a seed bank in the canopy that is released all at once into a post-fire environment uniquely favorable for lodgepole regeneration, mineral soil exposed by the removal of the litter layer, reduced competition from fire-killed plants, and the ashen-rich nutrient pulse that follows combustion of accumulated organic matter. The giant sequoia, Sequoiadendron giganteum, of California's Sierra Nevada carries this fire dependence even further into its evolutionary identity. Like the lodgepole pine, it maintains serotinous cones that open preferentially in response to fire-related heat and desiccation. But beyond seed release, the giant sequoia requires fire to prepare its seedbed. Sequoia seeds, which are among the smallest produced by any conifer relative to the adult tree's immense stature, germinate poorly in deep duff layers and accumulate litter, requiring the bare mineral soil exposed by fire for successful establishment. Furthermore, the thick, spongy, tannin-rich bark of mature giant sequoias, sometimes reaching 60 centimeters in thickness at the base, provides extraordinary insulation against the intense but typically brief heat of surface fires, allowing trees that have survived the first few centuries of growth to pass through subsequent fires with minimal damage, emerging from each event surrounded by the ash of competitors while themselves standing intact. The chemical ecology of fire-adapted forests reveals an evolutionary arms race of remarkable intricacy. Several plant species native to fire-prone ecosystems produce allelopathic compounds, substances toxic or inhibitory to competing plants, that persist in the soil and suppress the germination and growth of potential competitors during the post-fire establishment window. In the chaparral of California, species such as Adenostoma fasciculatum produce water-soluble phenolic compounds that leach from leaf litter into the soil, suppressing understory herbs and grasses during years between fires. Following a fire that removes the accumulated litter and temporarily deactivates these allelopathic compounds through volatilization and combustion, a brief window of extraordinary floral diversity opens in the chaparral, annual wildflowers, bulbs, and fire followers that have lain dormant as seeds or underground storage organs for years, sometimes decades, awaiting precisely this combination of released inhibition and exposed mineral soil. Smoke itself, long considered merely a byproduct of combustion, has proven to be a potent biological signal. Research initially conducted on South African fynbos species but subsequently extended to fire-adapted plant communities worldwide has demonstrated that many seeds from fire-adapted ecosystems require exposure to smoke, specifically, the compound carotenoid, a butanolide derivative produced during the incomplete combustion of cellulose, to break their physical or physiological dormancy. Carotenoid binds to receptors in the seed's embryo tissue and triggers a hormonal cascade that promotes germination, suggesting that these seeds have evolved not merely to survive fire but to be actively informed by it, to use the chemical products of combustion as a reliable signal that the disturbance event that will favor their establishment has occurred. The role of soil in forest ecology is so foundational and so multidimensional that treating it as a mere substrate entirely misrepresents its nature. A single teaspoon of temperate forest soil contains on the order of 1 billion bacteria, several kilometers of fungal hyphae, and a community of protozoa, nematodes, mites, springtails, and earthworms whose combined metabolic activity drives the decomposition of organic matter, the cycling of nitrogen, phosphorus, and sulfur, and the maintenance of soil structure in ways that no current agricultural technology has come close to replicating artificially. The organic horizon of an old-growth forest soil, the dark, humus-rich layer formed by centuries of accumulated, partially decomposed plant and animal material, represents the stored metabolic legacy of the forest itself, a biological memory encoded not in DNA or chemical signals but in the structural and chemical complexity of transformed carbon compounds. Nitrogen cycling in forests depends critically on the activity of specialized microorganisms that perform energetically demanding transformations invisible to any macroscopic observer. Biological nitrogen fixation, the conversion of atmospheric dinitrogen to ammonium ions, a form biologically accessible to plants, is performed in forest soils primarily by free-living bacteria such as Azotobacter and Clostridium, and by symbiotic bacteria of the genus Frankia living in the root nodules of actinorhizal plants such as alder, bayberry, and mountain mahogany. The energetic cost of biological nitrogen fixation is substantial, approximately 16 ATP molecules consumed per molecule of nitrogen fixed, and the nitrogenase enzyme responsible for the reaction is irreversibly inactivated by oxygen, requiring the organism either to maintain an anaerobic microenvironment or to produce protective oxygen-scavenging proteins. That evolution has solved this apparently contradictory problem, fixing nitrogen using an oxygen-sensitive enzyme in a predominantly aerobic environment, multiple times independently speaks to the extraordinary metabolic pressure that nitrogen limitation places on living systems. The mycorrhizal fungi that define the forest underground are not generic organisms but display remarkable host specificity and ecological specialization. Ectomycorrhizal fungi, the type that form a sheath around the fine root tips of trees rather than penetrating root cells, are predominantly associated with the dominant trees of temperate and boreal forests, oaks, beeches, birches, pines, spruces, and firs. Arbuscular mycorrhizal fungi, by contrast, penetrate root cells to form highly branched structures called arbuscules, the actual sites of nutrient and carbon exchange, and are associated with a vastly broader host range, including most tropical trees, many shrubs, and the majority of agricultural crop species. The two fungal guilds are not merely ecologically distinct but phylogenetically ancient in their separation. Ectomycorrhizal associations appear to have evolved independently many times from saprotrophic ancestral fungi, while arbuscular mycorrhizal fungi belong to an entirely different fungal phylum, the Glomeromycota, whose divergence from other fungal lineages occurred over 450 million years ago. Forest canopies, the uppermost stratum of mature forest vegetation, function as complex three-dimensional ecosystems in their own right, supporting communities of epiphytic plants, arthropods, birds, mammals, and amphibians that may never contact the ground surface across the entirety of their lives. The canopy of a temperate rainforest in the Pacific Northwest supports dozens of species of mosses, liverworts, and lichens that colonize the bark and branches of old conifers, intercepting fog drip, nutrient-laden throughfall, and airborne dust with surfaces collectively exceeding, in a mature stand, the total leaf area of the trees themselves. In the tropics, the diversity of the canopy exceeds that of virtually any other terrestrial habitat on Earth, with single trees in Amazonian old-growth sometimes hosting more arthropod species than are found in the entirety of the British Isles. A concentration of biological diversity so extreme that canopy biologists access their study sites by crossbow-fired ropes and aerial platforms, conducting research in what is effectively the least explored frontier in terrestrial ecology. The epiphytic bromeliads of Central and South American rainforest canopies constitute one of the most remarkable microecosystem architectures in nature. Each bromeliad, anchored to a tree branch by modified roots that function purely as holdfasts rather than absorptive organs, collects rainwater and organic debris in the central tank formed by its overlapping leaf bases, creating a miniature freshwater pond sometimes holding as much as 12 liters. Within each such phytothelm, a complete aquatic community assembles. Algae, bacteria, rotifers, ostracods, aquatic insect larvae, predatory damselfly nymphs, and in some species, entire populations of poison dart frogs that rear their tadpoles in these arboreal water bodies, carrying them individually up the forest giant from ground-level egg-laying sites to canopy cisterns where competition from aquatic predators is absent. The tree supports the bromeliad supports the frog consumes the invertebrates that would otherwise consume the bromeliad's algal community. A chain of mutual dependence assembled not by design but by the patient operations of natural selection across geological time. Old-growth forests, ecosystems that have developed over long periods without severe anthropogenic disturbance, typically defined as stands with trees exceeding 150 to 200 years of age in temperate zones and often far older in boreal or montane contexts, possess structural and ecological properties that secondary forests, however vigorously they regenerate, may require centuries to replicate. The defining characteristics of old-growth forest structure include multi-layered canopies with pronounced vertical complexity, large-diameter live trees with deeply furrowed bark and complex crown architecture, high densities of standing dead trees, or snags, and abundant fallen logs in various stages of decay. Each of these structural elements supports communities of organisms that cannot persist without it. Cavity nesting birds require snags of specific diameter and decay stage, and certain lichens and mosses colonize the bark of old, large-diameter trees so slowly that their presence reliably indicates centuries of biological continuity at a site. The coarse woody debris on the forest floor of an old-growth stand, fallen logs in progressive stages of decomposition, classified by ecologists using a 5-class system ranging from freshly fallen solid wood to moss-covered mounds of soft, red-brown humus, constitutes one of the most significant carbon pools in the terrestrial biosphere and one of the most biologically active. A single large fallen log of Douglas fir in the Pacific Northwest may require 300 to 500 years to fully decompose, supporting across the arc of its decay a sequential succession of fungal, invertebrate, and vertebrate species, bark beetles in the first decade, wood-boring longhorn beetles in the second and third, red-backed voles in the hollow interior of the later stages, and salamanders sheltering beneath the moist, spongy mass of nearly mineralized wood in the final centuries. The log does not merely die. It participates in the forest economy for longer as a dead structure than many trees live as standing ones. Carbon sequestration, the removal of carbon dioxide from the atmosphere and its storage in biological or geological forms, represents one of the most critical ecosystem services performed by forests, and one whose quantification has consumed decades of scientific effort. Forests globally store approximately 860 billion tons of carbon, roughly equivalent to all the carbon currently residing in the atmosphere, distributed among living biomass above and below ground, deadwood, litter, and soil organic matter. The majority of this carbon, roughly 44%, resides not in living trees but in the soil, a fact that dramatically complicates efforts to use forest protection and reforestation as climate mitigation tools, since soil carbon is both less visible than above-ground biomass and considerably more sensitive to disturbance, warming, and changes in moisture regime. The net carbon balance of any given forest, the difference between the carbon it sequesters through photosynthesis and the carbon it releases through respiration, decomposition, and disturbance, varies across the forest's developmental trajectory in ways that are ecologically intuitive but often politically inconvenient. Young, rapidly growing forests may be strong net carbon sinks, absorbing carbon from the atmosphere faster than they release it. Old-growth forests, by contrast, were long assumed to be carbon neutral, their photosynthesis matched by the respiration and decomposition of their massive accumulated biomass, but extensive recent research has overturned this assumption, demonstrating that intact old-growth forests continue to accumulate carbon net of all losses, acting as long-term sinks that young managed forests can take centuries to replicate even under optimal conditions. The carbon stored in an ancient forest is not only the carbon held in living wood but the carbon stabilized in old, deep soil organic matter that may have been accumulating for thousands of years. Tropical rainforests, despite covering only approximately 6% of Earth's land surface, are responsible for approximately 30% of terrestrial photosynthesis, a disproportionate contribution to global carbon and oxygen cycling that reflects their extraordinary year-round productivity in the warmth and moisture of the equatorial zone. The Amazon basin alone transpires so much water vapor into the atmosphere, roughly 20 billion tons per day, more than the daily discharge of the Amazon River itself, that it generates its own rainfall regime through the formation of what researchers have termed flying rivers, corridors of moist air that transport water from the Atlantic coast deep into the South American interior, sustaining rainfall patterns across the continent's agricultural heartland. This self-reinforcing moisture cycle means that deforestation of the Amazon does not merely remove trees but progressively dismantles the atmospheric engine that makes the remaining forest viable, with modeling studies suggesting the existence of tipping points beyond which large portions of the basin may convert to dry savanna through positive feedback mechanisms regardless of whether deforestation continues. The relationship between forest diversity and forest stability, the degree to which more species-rich forests are better able to maintain function in the face of disturbance, disease, or climate variability, has been one of the most actively researched questions in forest ecology over the past 3 decades. Multiple long-term experimental platforms, including the biodepth grassland experiments in Europe and the Cedar Creek biodiversity experiments in Minnesota, established with reasonable certainty that species richness positively influences primary productivity and community stability in grassland systems. Extending these findings to forests has proven more complex, because tree species diversity operates across spatial and temporal scales far larger than those accessible to controlled experimentation. But natural experiments, comparisons between monodominant tree plantations and structurally diverse natural forests under shared stresses, consistently suggest that diversity confers resilience through a combination of functional redundancy, niche complementarity, and the presence of rare species that become critical during unusual environmental conditions. Root systems constitute a forest's most extensive and in many ways most biologically active organ, yet their study has historically lagged far behind above-ground ecology for the straightforward reason that they are invisible, difficult to excavate without destruction, and their dynamics, root turnover, exudate release, interaction with soil organisms, occur on time scales that make continuous observation technically demanding. The total fine root biomass of a temperate forest, roots less than 2 millimeters in diameter, may exceed the above-ground herbaceous biomass several times over, and these fine roots turn over at rates of 1 to several times per year, meaning that the carbon investment represented by their continuous replacement constitutes one of the largest single fluxes in the forest's energy budget. The exudates released by living root tips, sugars, amino acids, organic acids, and phenolic compounds, constitute the primary carbon source for the enormous community of microorganisms that inhabit the rhizosphere, the zone of soil immediately surrounding and influenced by the root. The understory of a mature forest is not a degraded version of the canopy community but a distinct ecological zone shaped by conditions that the canopy creates rather than merely tolerates. Light levels on the forest floor of a closed canopy temperate forest may be as low as 1 to 2% of incident radiation, a deeply shade-adapted environment that nonetheless supports specialized communities of ferns, forbs, mosses, and shrubs whose photosynthetic apparatus has been optimized through evolution for maximum efficiency at low light intensities. The photosynthetic cells of shade-adapted understory species maintain chloroplasts of exceptional size and unusually high chlorophyll content, oriented in stacked grana of extraordinary depth to capture every available photon. Some understory plant species have evolved to exploit the brief but intense sunflex, ephemeral patches of direct sunlight that penetrate canopy gaps and move across the forest floor as the sun tracks through the sky, responding to a sunfleck that may last only seconds with rapid metabolic activation that would be energetically wasteful in a plant that experienced direct sunlight continuously. Among the most remarkable examples of chemical communication in forest ecosystems is the phenomenon of systemic acquired resistance in trees exposed to pathogenic attack. When a tree is infected by a fungal pathogen, bacterial disease, or viral agent, it does not merely mobilize local chemical defenses at the site of infection but may trigger a systemic response throughout the entire organism, a phenomenon mediated by long-distance signals transported through the phloem and xylem that include salicylic acid, jasmonic acid, and reactive oxygen species. These systemic signals prepare tissues distant from the infection site for potential attack, inducing the expression of pathogenesis-related proteins, cell wall strengthening enzymes, and volatile compound production that collectively reduce the pathogen's ability to colonize new tissues. The parallels with the vertebrate immune system, a systemic, coordinated, anticipatory response to threat that persists after the threat's resolution, are functionally compelling even if their molecular mechanisms evolved entirely independently. The strangler figs of tropical forests execute what is arguably the most dramatically strategic life history in the plant kingdom. A strangler fig, any of several dozen species in the genus Ficus, begins its life as a tiny seed deposited in the canopy of a host tree in the feces of a bird or mammal, germinating on an epiphytic perch of bark and accumulated debris with no connection to the soil far below. The seedling grows as an epiphyte for years, extending aerial roots downward along the host tree's trunk. As these roots reach the soil and begin absorbing water and nutrients, the fig's growth accelerates dramatically. The roots thicken anastomose, fusing where they contact one another, and gradually envelop the host trunk in an increasingly rigid, woody lattice. Over decades, the lattice tightens, potentially killing the host through a combination of hydraulic competition and mechanical compression, leaving the fig standing as a hollow column around the ghost of its former support, a tree that killed a tree to become a tree. The coevolution of figs and fig wasps represents one of the most ancient and obligate mutualistic relationships known in biology, estimated to be approximately 90 million years old, predating the mass extinction that eliminated the non-avian dinosaurs. Every fig species worldwide, of which there are approximately 800, is pollinated by one or a very small number of wasp species in the family Agaonidae, which breed exclusively within fig fruit. Female wasps enter the developing fig through a narrow channel called the ostiole, losing their wings in the process, to pollinate the fig flowers within and lay eggs in some of them. The wasp larvae develop within galled fig flowers, emerging as adults, wingless males and winged females, within the closed fig, mating in the dark before the males chew an exit tunnel through the fig wall, and the females, dusted with pollen from the fig stamens, depart to find another receptive fig in which to begin the cycle again. Neither the fig nor the wasp can reproduce without the other. Their fates are so thoroughly intertwined that the extinction of either partner would, in most cases, precipitate the extinction of the other. Temperate deciduous forests display a phenological choreography across the year that, observed in its full temporal sweep, possesses the structural elegance of a composition, a fugue in green and gold whose themes repeat with variation through centuries. The vernal awakening begins below ground, where root systems resume absorptive activity in response to soil temperature thresholds weeks before any above-ground change is visible. The cambium activates, producing a new flush of xylem and phloem cells. Then, in a sequence time to the precise interplay of accumulated heat units and photoperiod signals that varies by days or weeks between species, the buds burst. First the silver maples and red maples, before the canopy closes, then the oaks and hickories, calibrating their leaf out to the last frost dates encoded in their adaptive history, then the beeches and tulip poplars, completing the closing of the canopy and the transformation of the forest floor from a sun-drenched meadow into a deep green cathedral whose ceiling admits only fragments of sky. The senescence of deciduous leaves in autumn, the withdrawal of chlorophyll and the unmasking of yellow and orange carotenoids that had been present all season, obscured, plus the synthesis of red and purple anthocyanins that are genuinely new, not previously present, follows a molecular program no less precise than the vernal flush, triggered by shortening photoperiod detected by phytochrome photoreceptors in the leaf tissue rather than by temperature paresy. The adaptive logic of bright autumn coloration remains actively debated. One prominent hypothesis holds that anthocyanin production signals to aphids and other herbivorous insects that this tree is particularly well defended, the vivid color a costly signal of fitness that only the healthiest, most resource-rich trees can afford to produce in the brief window between senescence initiation and leaf fall. The honest signal hypothesis would predict that bright autumnal coloration correlates with better tree health and lower aphid infestation, a prediction that has received at least partial empirical support from comparative studies of co-occurring tree species. Water transport in trees defies casual intuition about how liquids move. In a tall tree, water must travel from roots to the uppermost leaves against gravity, a distance of 100 meters in the tallest known specimens, through a system of dead, hollow xylem conduits with no pumping mechanism analogous to a heart. The mechanism, the cohesion tension theory, depends on the extraordinary tensile strength of liquid water under negative pressure. As water evaporates from leaf surfaces through stomatal pores in the process of transpiration, the resulting tension is transmitted through the continuous water column in the xylem all the way to the soil, pulling water upward against gravity through what is essentially a very long, thin straw. The water in the xylem of a tall tree is under considerable negative pressure, effectively in a metastable state of tension that, if disrupted by an air bubble entering the xylem conduit, can cause embolism, the formation of a vapor pocket that breaks the water column and can, in severe cases, contribute to hydraulic failure and tree death. The stomata of leaves, microscopic pores flanked by pairs of guard cells that regulate their aperture, are the site of a continuous negotiation between the leaf's need to absorb carbon dioxide for photosynthesis and its need to limit water loss to transpiration. When water is plentiful and light is abundant, guard cells accumulate potassium ions through active transport, increasing their osmotic potential and driving water influx by osmosis, swelling them and opening the pore between them. Under drought stress, falling leaf water potential triggers the synthesis and release of abscisic acid, a phytohormone that causes guard cells to lose their turgor by releasing potassium and triggering the efflux of water, closing the stomata and dramatically reducing transpiration, but simultaneously reducing carbon dioxide uptake and photosynthetic carbon fixation. The tree in drought is thus faced with a physiological dilemma: close stomata to preserve water and risk starvation, or maintain stomatal aperture to fix carbon and risk desiccation. The resolution of this dilemma varies by species in ways that reflect evolutionary adaptation to the moisture regimes of their native ranges. Forest sounds. The creaking of limbs in wind, the percussion of woodpeckers, the murmur of running water, the rustle of small animals through leaf litter, compose an acoustic environment whose complexity has only recently become the subject of systematic scientific study. The field of soundscape ecology, pioneered by researchers including Bernie Krause and the team at Purdue's Center for Global Soundscapes, examines the relationship between the acoustic signatures of ecosystems and their ecological integrity. Healthy, biodiverse forests produce rich, continuous soundscapes in which different species partition the acoustic spectrum, some vocalizing in low frequencies, others in high, some at dawn, others at dusk, in arrangements that collectively represent the community's history and health. Disturbed forests, by contrast, produce impoverished soundscapes with large temporal and spectral gaps, silences that Krause has termed geophony, biophony, and anthrophony, distinguishing the sounds of wind and water from those of living organisms and those of human technology. The soundscape in this framework is a diagnostic. Listen to a forest and you are reading its ecological report card. The redwood forests of California's northern coast are among the most structurally complex and biomass-dense ecosystems on Earth, supporting above-ground live biomass exceeding 2,600 tons per hectare in the most productive old-growth stands, a figure that exceeds that of any tropical forest by more than 3 times. The trees responsible for this extraordinary accumulation, Sequoia sempervirens, achieve their record stature. The tallest measured individual, Hyperion, stands at 115.9 meters, through a combination of rapid early growth rates made possible by the mild, moist climate of the fog belt and exceptional longevity made possible by bark chemistry and wood density that confer near immunity to insect attack and fungal decomposition. The fog itself, advective fog formed when warm Pacific air moves over the cold California current and condenses, is not merely an atmospheric phenomenon to these trees but a direct water resource, intercepted by the fine foliage of the upper canopy and dripping as fog drip to the forest floor at rates that can exceed rainfall during the dry summer months, sustaining the forest through the Mediterranean dry season, without which the redwood's year-round growth would be impossible. The Pacific salmon that spend their adult lives in the open ocean but return to the freshwater streams of their birth to spawn and die constitute one of the most remarkable vectors of marine nutrient subsidy to terrestrial ecosystems known in ecology. Salmon carry in their bodies nitrogen and phosphorus derived from marine food webs. Stable isotope analyses reveal the marine signature in this nitrogen as an elevated ratio of nitrogen-15 to nitrogen-14, a signal traceable through the food web, and as they die in stream channels, are consumed by bears, otters, eagles, and ravens, and their carcasses decompose into the riparian soil. They deliver this marine-derived nitrogen to the trees growing along the stream banks. Studies in Alaska and British Columbia have documented marine nitrogen in the foliage and annual rings of riparian Sitka spruce growing beside salmon streams, with trees close to productive salmon streams growing significantly faster than those beside streams without salmon runs. The forest does not merely surround the stream. It is, in part, made of the ocean, assembled one salmon at a time across centuries of migration and death. The evolution of seed dispersal syndromes, the co-evolved suites of fruit characteristics and disperser traits that have emerged between plants and the animals that move their seeds, represents one of the most intricate and beautiful examples of coevolution in the forest. The fleshy, colorful fruits of forest trees are not biological accidents but advertisements, shaped over millions of years of selection to appeal to the sensory systems and dietary preferences of specific animal dispersers. The red and black fruits of elderberry and pokeweed attract passerine birds with color vision sensitive to short wavelengths. The rich lipid content of fruits consumed by migratory birds provides the concentrated energy needed for long flights, while the seeds pass through the bird's digestive system intact and are deposited, sometimes hundreds of kilometers away, in the coating of nitrogenous droppings that serves as a germination stimulant. Some seeds, including those of several tropical trees, require passage through the gut of a large mammal to physically or chemically scarify their hard seed coats before germination becomes possible, making the tree's reproduction not merely facilitated by animal partners but absolutely dependent on their continued existence. The endophytic fungi and bacteria that live within the tissues of living, healthy trees, not as pathogens but as persistent, largely benign residents of leaf, bark, and wood tissue, constitute a largely hidden dimension of forest biology whose ecological significance is only beginning to be appreciated. Endophytes, estimated to be present in virtually every plant species examined in sufficient detail, colonize plant tissues through leaf stomata, lenticels, and root surfaces and then persist without causing visible disease symptoms in a state of apparent mutualistic or commensalistic balance with their host. Some endophytes produce secondary metabolites that function as chemical defenses against herbivores and pathogens, effectively extending the plant's defensive repertoire at the cost of providing the endophyte with carbon and shelter. A single leaf may harbor dozens of endophyte species, and the community composition varies predictably between tree species, between sun-exposed and shaded leaves on the same tree, and across the seasons, suggesting that endophyte communities are not random colonists but structured, regulated, dynamically maintained associations whose ecological importance to forest tree health may prove comparable to that of the mycorrhizal network below ground. Forest fragmentation, the division of continuous forest habitat into smaller, isolated patches by roads, agriculture, and urban development, affects forest ecosystems through mechanisms that extend far beyond the simple arithmetic of area reduction. Edge effects, the ecological changes associated with the transition zone between forest and non-forest habitat, penetrate deep into forest fragments, altering temperature, humidity, wind exposure, and light levels for distances of 100 meters or more from the fragment boundary. In a small forest fragment, the area genuinely insulated from edge effects may represent a fraction of the total area, meaning that the ecological character of the fragment is determined more by its periphery than its interior. Large mammals, birds with large territory requirements, and plants dependent on interior forest conditions may be functionally absent from fragments that appear, viewed from above, to provide adequate habitat. The extinction debt, the future species losses that are predicted to occur as populations in fragmented habitat decline to below minimum viable population sizes, a process that may play out over decades or centuries after the fragmentation event, means that the full ecological consequences of current forest loss will not be apparent until long after the habitats that would reverse it have been converted. Mangrove forests, the salt-tolerant, tidally inundated forests that fringe tropical and subtropical coastlines on every continent, represent one of the most physiologically and structurally extraordinary forest types on Earth, simultaneously managing the challenges of salt exclusion, anaerobic waterlogged soils, wave energy, and tidal fluctuation in an environment that would be immediately fatal to almost any other tree species. Mangrove trees have evolved a remarkable diversity of solutions to these challenges. Some species exclude salt at the root surface through ultrafiltration mechanisms so selective that the xylem sap is nearly salt-free. Others secrete excess salt through specialized glands on their leaf surfaces, producing visibly crystalline deposits on a sunny morning. Their root systems display extraordinary architectural diversity, prop roots descending from above-ground stem tissue, pneumatophores projecting vertically upward from the waterlogged substrate like a field of snorkels to conduct oxygen to anaerobic root tissues below, each architecture representing a different evolutionary solution to the challenge of living in an environment where soil oxygen is essentially absent. Tropical montane cloud forests, forests that grow at middle elevations on tropical mountain slopes where persistent cloud immersion provides a near-constant supply of moisture, host some of the highest plant species diversity of any terrestrial ecosystem, including extraordinary concentrations of epiphytic orchids, bromeliads, ferns, and mosses that clothe every available surface of the forest in layers of emerald and silver green. The cloud immersion these forests depend upon is maintained by the interplay of regional topography, ocean temperatures, and atmospheric circulation patterns, making them exquisitely sensitive to climate change. As warming shifts the elevation of cloud formation upward, montane cloud forests may find their fog zone migrating above the forest canopy, effectively stranding the community of cloud-dependent organisms in an increasingly drought-stressed environment. The extinction risk associated with upward cloud migration is particularly severe because montane species are already confined to a narrow altitudinal band and have no higher elevations to retreat to when conditions change. The bark of trees is a biological archive of threats, inscribed in the language of chemistry and structure. The outer bark, or rididome, is composed of dead, suberized cells whose function is primarily protective, resisting desiccation, deflecting the boring attempts of insects, and insulating the living cambium against temperature extremes and pathogenic invasion. Beneath it, the inner bark, composed of living phloem cells and parenchyma, is the site of the tree's chemical defenses, resin canals in conifers that are maintained under positive pressure and discharge sticky, polymerizing terpene mixtures when breached, latex systems in trees such as rubber and euphorbia that release viscous, toxic fluids, polyphenol-rich tannin cells in oaks and chestnuts that constitute a diffuse defensive chemistry throughout the bark tissue. The thickness, texture, and chemistry of bark represent the accumulated evolutionary response of each tree species to the particular community of bark beetles, wood-boring insects, mammalian herbivores, and fungal pathogens that have coexisted with it through evolutionary time, a material record of ancient antagonisms written in cork. Among the chemical compounds produced by trees for defensive purposes, the terpenoids represent the largest and most structurally diverse class, encompassing monoterpenes, sesquiterpenes, diterpenes, and triterpenes synthesized through the mevalonate and methylerythritol phosphate pathways from simple acetyl-CoA precursors. The monoterpenes, alpha-pinene, limonene, myrcene, and dozens of relatives, are responsible for the characteristic scents of pine and fir forests, released from resin glands, crushed needles, and wounded bark into the warm summer air where their volatility ensures rapid dispersal. These compounds function simultaneously as toxins to bark beetles and their fungal symbionts, as attractants to the parasitoids and predators of those beetles, and, when released in sufficient quantities into the atmosphere, as precursors to secondary organic aerosol particles that contribute to the characteristic blue haze above forested mountains. The forest, in short, is painting its own sky, using the chemistry of defense to generate the optical phenomena that have given the Blue Ridge and Smoky Mountains their names. The evolution of broad-leaved deciduous trees in response to the seasonal temperate climate represents a trade-off solution of remarkable elegance. The flat, thin blade of a deciduous leaf, maximizing the surface area available for light capture and gas exchange during the warm growing season, is simultaneously highly vulnerable to freezing damage, physical tearing by ice and wind, and the cumulative costs of maintenance and defense across the full annual cycle. The deciduous strategy accepts these vulnerabilities by discarding the entire leaf at the onset of the unfavorable season, at the cost of the photosynthetic investment it represented, but recovers the most valuable molecular components first. Nitrogen from chlorophyll proteins is remobilized and stored in stem and root tissue before abscission, and begins the next season with a fresh leaf, synthesized from the previous year's stored nutrient capital. The evergreen strategy, maintained by conifers and many tropical trees, retains the leaf across multiple years, spreading the construction cost across a longer productive lifespan but maintaining a leaf that must be more heavily defended against herbivory and more robust against physical damage, and whose photosynthetic rate per unit area tends to be lower as a consequence of the structural investments that longevity requires. Bioluminescent fungi, species within the Mycena, Pinellus, Omphalodus, and Neonothoponus genera, among others, produce a dim, continuous greenish-blue light through the oxidation of a luciferin compound by a luciferase enzyme in a reaction requiring molecular oxygen. The ecological function of fungal bioluminescence remains one of the more genuinely mysterious questions in forest biology. Unlike the bioluminescence of fireflies or deep-sea organisms, which has been convincingly linked to mating signals or predator attraction, fungal bioluminescence operates at intensities and in ecological contexts that make its adaptive significance far less obvious. One hypothesis holds that the light attracts nocturnal arthropods that disperse fungal spores, analogous to the way flowers use visual signals to attract pollinators. Laboratory experiments have provided some support for this idea, demonstrating that spore dispersal from bioluminescent mycelia is significantly higher in the presence of artificially simulated bioluminescence. If confirmed in field conditions, this would represent a convergent evolution of optical signaling in organisms that separated from a common ancestor more than 1 billion years ago, the forest floor lit, across the deep watches of the night, by chemical lamps whose purpose is to ensure that the network persists. The growth of tree roots toward water sources, hydrotropism, was for centuries attributed simply to water diffusion gradients, but more recent research has revealed a more sophisticated picture involving the active sensing of water potential gradients by root cap cells and the asymmetric redistribution of the hormone auxin to mediate differential growth on the wetter and drier sides of the root tip. Root cap cells contain amyloplasts, starch-filled organelles that sediment in response to gravity and serve as gravity sensors in gravitropism, and appear to perform an analogous sensing function for water potential gradients, translating differences in turgor pressure across the root cap into directional growth signals. Roots navigating toward water in heterogeneous soils do not blunder toward it by chance but are guided by a sensory system of considerable discrimination, capable of steering through substrates of varying texture, chemistry, and moisture content with a precision that, observed under time-lapse photography, appears purposeful in a way that challenges our common-sense assumptions about the boundary between awareness and mechanism. Phenological mismatch, the disruption of timing relationships between interacting species caused by differential responses to climate warming, represents one of the more insidious ecological threats posed by rapid climate change to forest ecosystems. Many of the critical ecological interactions that maintain forest community structure depend on precise temporal synchrony, the hatching of caterpillar larvae timed to coincide with the peak nutritional quality of emerging tree leaves, the flowering of forest herbs timed to coincide with the activity windows of their specialized pollinators, the fruiting of mycorrhizal fungi timed to coincide with the period of maximum carbon allocation from tree roots. When the temperature sensitivity of each partner's phenology differs, as it almost always does, because each species has its own thermostat calibrated by its own evolutionary history, warming may advance the phenology of one partner while leaving the others unchanged, creating a temporal gap where a temporal overlap once existed. The forest's carefully tuned ecological clock is being reset by a climate that is changing faster than the evolutionary recalibration of species interactions can accommodate. The response of forests to elevated atmospheric carbon dioxide, a question of obvious urgency given the trajectory of fossil fuel emissions, has been examined through free air carbon dioxide enrichment experiments, or FACE experiments, in which sections of intact forest are exposed to elevated CO2 concentrations using a ring of pipes that release carbon dioxide into the forest air. These experiments, conducted in temperate forests in the eastern United States, boreal forests in Sweden, and tropical forests in Australia, have consistently shown that elevated CO2 stimulates photosynthesis and initially increases growth rates, the carbon fertilization effect, often cited by those skeptical of forest vulnerability to climate change. However, longer-term monitoring of FACE experiments has revealed that this growth stimulation is typically transient, declining over years as other nutrient limitations, particularly nitrogen and phosphorus, constrain the forest's ability to translate additional carbon availability into biomass. The forest, in other words, cannot simply eat more CO2 without a corresponding increase in the other nutrients needed to build wood, and those nutrients are, in most of the world's forests, already in short supply. Mosses, bryophytes lacking vascular tissue, roots, and, in most species, any anatomical structure for conducting water or nutrients over more than the most minimal distances, are among the oldest land plant lineages on Earth and among the most ecologically consequential for forest function despite their diminutive stature. A continuous moss layer on the forest floor performs insulating, moisture-retaining, and decomposition-mediating functions whose cumulative importance to the thermal and hydrological regime of the soil below vastly exceeds what the moss's modest biomass would suggest. In boreal forests, the feather moss carpet, formed primarily by Pleurozium shrubbery, Hylocomium splendens, and Sphagnum species, insulates the permafrost soil from summer warming with a thermal resistance that maintains frozen ground temperatures in the face of air temperatures that might otherwise thaw the permafrost and release the enormous quantities of methane and carbon dioxide stored within it. The moss, in this context, is not merely a ground cover but a climate regulator, a living thermostat that has been performing its function continuously since the boreal forest first established following the retreat of the Pleistocene ice sheets. Sphagnum moss, the peat-forming genus responsible for the formation and maintenance of bogs across the boreal and subarctic zones, has shaped the carbon chemistry of the northern hemisphere to a degree that few organisms outside of marine phytoplankton can match. Sphagnum produces an extraordinarily recalcitrant polymer, sphagnin, that acidifies its environment by exchanging hydrogen ions for metal cations from the surrounding water and inhibits the activity of decomposer microorganisms, causing the organic matter it produces to accumulate rather than mineralize. Over thousands of years, in waterlogged depressions where sphagnum growth keeps pace with or exceeds decomposition, the accumulated biomass forms peat deposits whose depth may exceed 10 meters and whose age may span the entire Holocene. The northern peatlands collectively store approximately 500 billion tons of carbon, comparable to all the carbon in the atmosphere, sequestered in the cold, acid, waterlogged conditions that sphagnum creates and perpetuates. The fate of this carbon under warming conditions, where thawing and drying of peatlands could reverse 10,000 years of accumulation within decades, represents one of the most consequential and least tractable risks in the global carbon budget.